Hello everyone, and thank you for being here today to listen to my presentation entitled Zoonotic Pathogens in Broiler Chickens, Working at the Animal and Human Interface. I'm Marie-Lou Gaucher. I'm part of the Research Chair in Meat Safety at the University of Montreal, the Research Chair in Meat Safety that was founded in 1999 by Dr. Sylvain Kissy. Our research group is now composed of 4 different researchers, namely Sylvain Kissy, Alexandre Thibodeau, Mohamed Rouma, and myself. The expertise of our group focuses on food and especially meat safety. On zoonotic pathogens, we study their epidemiology and control from the farm to the fork. We are also interested in host pathogen and interactions, as well as in the microbial ecology of meat, eggs, production surfaces, and from the digestive tract of food-producing animals. We are also experts in risk analysis. My research program focuses on sustainable poultry production and food safety, and I'll walk you through the different areas of this research program that are sustainable poultry production, studying Clostridium perfringens, and working at the animal and human interface with foodborne pathogens that are Salmonella and Clostridium perfringens. So let me give you a brief overview of my research program. Why talking about sustainable poultry production? Because antibiotic resistance is a driving force behind the transition away from routine use of antibiotics in poultry production. And as many of you might know, the different antibiotic categories have been withdrawn over the past few years, leaving very few molecules for poultry production and making poultry production a bigger challenge. We started a few years ago by evaluating the impacts of rearing broiler chickens without using antibiotics. So on 8 different broiler chicken farms in Quebec during 15 months, we conducted a study comparing a conventional treatment using antibiotics and anticoccidials to a drug-free program not using those molecules and replacing them by alternative natural oil-based products, an acidification of the drinking water, and a coccidiosis vaccination strategy. So from these 8 participating farms, 2 farms withdrew from the study, 4 farms did a short-term, so 15-month long antibiotic withdrawal, and 2 farms after the study never reintroduced antibiotics. So what we observed, we published a scientific paper, but at the farm level, we observed that the drug-free program was associated with a decreased mean daily weight gain, an increased feed conversion ratio, an increased necrotic enteritis incidence, and this study also allowed us to build a unique collection of C. perfringens strains from which we conduct great research projects today. From a public health perspective, we were interested in documenting the impacts of rearing antibiotic-free broilers on the problem of antibiotic resistance. So at the end of the study in 2012, as well as in 2018, we collected from these participating farms the CICA from 12 broilers, and we quantified the relative abundance of 7 antibiotic resistant genes encoding resistance to bacitracin, ptilocin, virginamycin, lincomycin, and sulfase. We published these results last year, but what we observed was that a long-term antibiotic use was associated with an increased abundance of antibiotic resistance genes. A short-term antibiotic withdrawal had no significant impact on the presence of those genes, and a long-term antibiotic withdrawal was associated with a significant decrease in the abundance of those antibiotic resistance genes. We are now working at the animal and human interface, and we are now conducting a research project evaluating the contribution of bioaerosols to the exposition of workers and communities to antibiotic resistance genes. So we are currently conducting visits on broiler chicken farms using or not using antibiotics, as well as in poultry slaughter plants in Quebec and in Saskatchewan. During these visits, we perform air sampling inside the facilities and outside the facilities to document the exposition of workers and communities around these facilities to ARGs. And using a high throughput sequencing approach, we will document the nature and abundance of these resistance genes, as well as the bacterial populations that we can find. This brings us to studying Clostridium perfringens. As I mentioned, the field study conducted a few years ago allowed us to build a unique collection of C. perfringens strains, and we characterized this collection in part as we wanted to know what was the impact of experiencing necrotic enteritis or not experiencing the disease on the C. perfringens population richness dynamics over time, and we reported that for healthy barns not experiencing necrotic enteritis, the C. perfringens population richness remains the same over time, whereas experiencing necrotic enteritis has a profound impact on the richness of the C. perfringens population, and after a certain time we can observe that the virulent clones take all the place and dominate the C. perfringens population in these barns. So we concluded that in drug-free flocks, there might be a competitive advantage for any causing C. perfringens, and this led us to work on a new research project in which we wanted to identify putative adhesins unique to any causing C. perfringens using a comparative and subtractive reverse vaccinology approach. And this project has been going on in our lab since 2017. A first scientific paper was submitted last month in which we report the description of the C. perfringens proteome as well as the identification of 6 putative immunogenic adhesins unique to any causing strains. The last steps of this project are currently ongoing in our lab, during which we will evaluate the immunogenic and protection potential of these adhesins. As some of you might know, Clostridium perfringens is also a zoonotic pathogen, as some strains of this population can produce an enterotoxin encoded by the CPE gene. This enterotoxin is responsible for gastrointestinal illness, and different sources of exposition for humans have been reported, such as beef, poultry, and pork meats, vegetables, and fruits, but the reservoirs and routes of transmission remain largely undescribed today. And we were interested in documenting the role of poultry meat in the exposition of consumers in Canada. As you can see, enterotoxigenic C. perfringens ranks second among the most important foodborne pathogens in Canada. So we conducted a study in 2 different commercial slaughter plants in Quebec involving 79 broiler chicken flocks and nearly 400 carcasses that were sampled through a carcass rinse approach. Globally, what we found, and we published these results, was that 27% of the flocks sampled were positive for the presence of CPE. Up to 10% of the flock sample were positive to CPE upon arrival to the slaughter plant. 10% of the carcasses sampled overall were positive for CPE. Positive carcasses were identified at every step of the slaughter process, and up to 25% of the carcasses sampled after chilling were positive for the enterotoxin encoding gene. We are now conducting a new study that aims at describing better the epidemiology of enterotoxigenic Clostridium perfringens along the broiler chicken production chain. This has been ongoing since 2020. This is a 2-phase field study. A first sampling phase was conducted end of 2019, beginning of 2020. It involves 30 broiler chicken farms, and 2 visits per farm are conducted, 1 visit at chick placement and another visit conducted at the end of the rearing period. We collect different samples representing the contribution of breeder flocks, hatcheries, broiler farms, feed, transport, and slaughter plant. And the last phase of this project will begin 2022, and we are now isolating and characterizing the enterotoxigenic C. perfringens strains recovered. Same thing for Salmonella. It is, as you know, among the most important foodborne pathogens in Canada, and poultry is a significant contributing source for the exposition of consumers to Salmonella. As we did for enterotoxigenic C. perfringens, we also looked at the presence of Salmonella during the study conducted at the slaughter plant level. A scientific paper was published, but what we report in terms of results is that as much as 90% of the incoming birds were positive for Salmonella. The carcass status for Salmonella at the beginning of the slaughter process greatly impacts the final carcass status, as when a positive flock positive flock comes into the slaughter plant, chances are that 45% of the poultry meat products will end up being Salmonella positive, whereas when a negative flock comes into the slaughter plant, as less as 15% of the poultry meat products will be found positive for Salmonella at the end of the slaughter process. We also found that the flock status greatly impacts the status of the flock subsequently slaughtered on the same day. And we also found that using a high-resolution melting approach for typing the Salmonella strains recovered, we were able to demonstrate that some HRM types are more able to persist throughout the slaughter process. At the farm level, we are now conducting this same study for Salmonella, collecting sample at every different critical steps of the poultry production chain, and hopefully we will be able to have a better idea of the transmission dynamics of Salmonella along the broiler chicken production. Of course, this research wouldn't be possible without our precious industrial partners, as well as without the major funding organisms, but also without our precious collaborators that are from Quebec, Canada, United States, and from France as well. Well, thank you so much for your attention. I will be pleased to answer your questions.
Great. Well, thank you, Marie-Lou. If you want to just turn your mic off and join us on camera. Thank you for your presentation. Again, if you have any questions, please put them in the question and answer. So the first question is, have you done any analysis on zoonotic potentials of E. coli?
No, we did not look at E. coli.
Oh, is that anything that you might be interested in doing?
Well, we were not planning looking at E. coli for the coming phase of the on-farm project that I was describing, but we do have the samples, so that might be a good idea. Sure.
For sure. All right.